Hi everyone, I hope you can all hear me. Welcome to the webinar for tonight, the Australian Medicinal Cannabis Research as our topic, and it's hosted by myself. I'm Dunya, I'm one of the pharmacists here that works at Health House, and we've had a massive amount of interest in this subject with lots of exciting things happening at the moment. Just remember that tonight, the guidance that you might hear or the medical advice, we always prefer that you speak to your medical professional first before you follow any of that guidance. So it's not intended as a substitute for your current medication management plan. So there's 6 researchers tonight that we will be speaking to this evening, and it's a panel style questionnaire at the end. If you have any questions, at the bottom of your screen, you'll see a couple of options. Don't use the chat function because we don't want to just have a good old chat tonight. We want to answer some really interesting questions. So you can use that question and answer function at the bottom of your screen. And we'll try and get through as many as we can for about half an hour near the end. One thing we have to consider as well is around the world, the US normally leads the way with pharmaceutical research. But in this case, medicinal cannabis is currently federally illegal in the US, and research for them is really difficult. There's about 10 separate permits that they require and a lack of GMP produced concentrates. And looking at the research a little bit globally as well, the other limitation is much of the trials conducted have used smoked cannabis, so it makes it variable in quality and difficult to create a placebo option as well for patients. If we look at our homeland, Australia has a lot of advantages, and not the least of which is government support. Our Federal Health Minister, Greg Hunt, is really supportive of research. And there's lots of government grants and funding, especially in New South Wales and Victoria, where the state governments have been proactive in this space. And lastly, Australia has made sure that any cannabis used on our domestic market is of GMP quality. And this is overseen by the TGA, or the Therapeutic Goods Administration. So it's important to do the research properly without bias and test against placebo and/or other medications if it's possible, which is one area that the team here at Health House really does support. So enough of me. Let's start off with our first speaker. This is Professor Marco Falasca, who will join us near the end. He's from Curtin University here in WA. And he's been engaged in the development and the progression of pancreatic diseases, particularly discovering possibly new mechanisms and options for one of the most lethal types of cancer, being pancreatic cancer. You can see some of the impact this has with a couple of statistics that I've listed here happening in Australia. Marco and his team specialise in cell metabolism, and from 2018 have discovered potentially new medicinal cannabis treatment. He's currently testing a range of cannabinoid-based formulations in vitro and against human pancreatic cancer cell lines grown in the lab. So this is either standalone medicinal cannabis treatment or in combination with existing chemotherapy agents. His plans then include implementing these findings in human trials. So let's hear some of the things he has to say about his research.
Okay, we're recording. So first up, Marco, welcome. Thank you for joining us today and being part of this. How long have you been studying cannabis, and what first got you into this industry?
Thank you for giving me this opportunity. So I have a long-standing interest in lipid metabolism, biology, and in particular a class of lipid that is very similar to endocannabinoids. So in that sense, I've been interested in this lipid with the endocannabinoid structure and properties. But I think since the discovery of the receptor of this lipid in 2007, and this receptor it's a putative cannabinoid receptor. So this introduced me fully in this field. So and therefore start investigating cannabidiol first and then cannabis. So let's say it's more than 10 years now.
Wow, okay. And I'm really excited by your work on pancreatic cancer. Are you considering human trials?
Yeah, of course, our aim and objective and ultimate desire is to bring what we found in the lab setting and animal testing in trials. There are quite a few years we considering this option, but there are few obstacles that we need to overcome. So it's I hope we can get to trials quite soon, because we need a definite demonstration that the cannabis component, cannabinoids, can have a value and at least improve chemotherapy in pancreatic cancer settings.
I know there's plenty of people waiting for that work. So in your opinion, could a different ratio of THC and CBD or the minor cannabinoids potentially cure pancreatic cancer? I know you've extended the life of rat models, but do you think there's a cure there?
Cure is a big word. So especially in pancreatic cancer, we need to be very careful. It's a very aggressive disease. And we've seen good response, but I would be, a bit careful in talking about cure. So of course, this is a long-term aim, but definitely I think there is a great potential. Our data are demonstrating, at least in some patients. And this is a bit the complication we had, because we started investigating cannabidiol, and then the question was, is cannabidiol or is cannabis or other components synergizing with cannabidiol? And this is, of course, part of the research that is ongoing. So we have already some result. Hope we can publish this soon, so we can have a definite answer to this question, in at least in pancreatic cancer. But I think there is a value. This is what we're investigating: different cannabinoids combinations, and beyond pancreatic cancer, because we're now interested in other fields like obesity, diabetes, so inflammatory bowel disease. So there are many other conditions where this is well known, but we are interested in investigating these other fields as well.
Excellent. Thank you so much for your work and being part of this webinar. I really appreciate it.
Thank you.
Alrighty, so our next speaker for tonight is Dr. Amanda Timler, and she works at the University of Notre Dame here in WA, and she's researching the potential benefits of medicinal cannabis in dementia patients. So the trial she's part of uses a randomized double-blind crossover placebo control, and it's designed to evaluate the behavioral changes, quality of life, and the level of discomfort caused by pain in about 50 participants. This trial uses an oral spray, and it administers a combination of THC and CBD under the tongue, and in addition to the patient's current medication management plan. Let's hear what Amanda had to say about the trial findings so far.
So I'll go through a couple of questions with you. First one is, in what dementia symptoms are you looking for improvement part of your trial?
Yeah, so we're looking for a lot of the behavioral symptoms associated with dementia. So it's things like anxiety, maybe some things like aggression. So whether it's physical aggression, non-physical aggression, verbal aggression, that sort of thing. Maybe also improvements in sleep, as well as agitation and any forms of anxiety and depression. We're also looking at quality of life and pain, as well as two other factors.
Yeah, that's quite important as well, particularly for a lot of the dementia patients. What are some of the challenges that you've faced since starting, particularly with this lovely pandemic all hitting us at the beginning of the year?
This project has not come without its challenges.
Yeah.
COVID has not made it any easier in that regards. Right from the very beginning, there's been issues with legislation. So the Guardianship and Administration Act of 1990 has been silent on medical research. So it's really impacted on who we can actually include in the study. So trying to get the right participants. Initially it limited who we could include. So we could only include those with mild dementia, which is a bit of a shame because we think those with more moderate to severe symptoms would benefit the most from the medication. So it's caused us to go back and rework the project a little bit, shorten the duration, as well as make sure we look at the inclusion-exclusion criteria, make sure we're actually trying to get those who could benefit the most from the medication. We've also had issues with trying to get into aged care facilities. So the high turnover with staff has been quite stressful. From my perspective, that they don't tend to stay in the roles very long. So once I go in and engage with an aged care facility, by the time we're ready to administer or look at potential participants, they've kind of moved on to another role. Yeah, and then once they've
tricky, yeah. Particularly passing the information then to the next staff member, I bet.
Yeah, it's constantly re-engaging. Yeah, there's no sort of handover. There's no sort of thing. So I kind of am emailing them out of the blue, so to speak. Yeah.
You are the handover. Yeah. Okay.
Yeah.
And then what sort of effects did you find patients have, or their carers or family members are reporting so far in the trial?
Yeah, we're seeing that their family members seem to be a lot calmer, especially during COVID. It's made them their family members more relaxed, so it's kind of taken the pressure off them because they haven't been able to go and visit their family members in the facilities. They feel like them being on the medication has kind of helped with that in some regards. Yeah, there's also been sort of improvements with the lack of calling out, you know, more sense of relaxation, just that's sort of overall less agitated. So there's a lot of sort of positive stuff that we're starting to see.
Okay.
Even though it's not great evidence. Yeah.
Okay. And do you have anything you wanted to add? I guess I asked you certain ones that I would have liked to know about the trial, but if you feel like you want to add in something, go for it.
I guess the only thing is that the one positive that's come out of COVID is that they've actually changed the Guardianship and Administration Act to include medical research. So now we're going through and changing our approvals again to make sure we can include those with moderate and severe dementia. So yeah, we're now at the stage we wanted to be at the beginning.
That's awesome. Would that broaden as well the number that you're including part of the trial then?
We're still keeping with our initial number of participants.
Okay.
Yeah, but it's going to be a bit easier. Yeah, instead of going to the facility and only being able to recruit 2, we now might be able to go to the facility and recruit 10. So,
okay, cool.
Yeah.
Oh, awesome. Thanks so much.
Alrighty. So our next speaker for tonight actually began some of her work in 2018. She was part of the world first study at UWA, and initially the trial wanted to evaluate the safety and efficacy of using oral medicinal cannabis extract compared to a placebo for the treatment of insomnia. So Dr. Jennifer Walsh was part of a trial that administered sublingually at night, an hour before bed, in addition to the patient's current medication plan. And she'll explain now what else it entailed and what the team discovered at the time.
So according to the results available, we see that there was a decrease in the insomnia severity index by average of 26% across all participants and 36% with those treated with higher dosing. Could you please tell us a bit more about the results and if you were surprised with the outcome?
Yeah, well, actually, we were surprised that the results were as positive as they were. The participants had reductions in their insomnia symptoms, when they were taking the active medication versus the placebo. They reported sleeping longer, they were awake for less time during the night, and they felt more refreshed when they woke up in the morning. This data aligned with our objective data as well of their sleep quality. So we used actigraphy to measure their sleep quality and quantity. And so that's actigraphy based on a wrist-worn device that measures activity and inactivity, being a surrogate for sleep. And we measured this over 2 weeks. And the actigraphy data showed that they slept for longer and they woke up less during the night as well. And I must admit that, that one of the other things that I was quite surprised at is that despite going to great lengths to ensure that the placebo was very well matched to the active medication in terms of smell, appearance,
Right.
odour, for example, every single one of the participants actually identified when they were taking the active medication. So, and it wasn't because they could see a difference or smell a difference or taste a difference, it was because of the effect of the medication, that they slept better. So obviously you imagine that it's very difficult to blind for that effect.
Yeah. Wow.
Yeah.
Interesting to know. What were some of the inclusion and exclusion criteria used in your study?
So we took men and women who were aged between 25 and 70 years, and they had to have chronic insomnia. And we defined this as self-reported difficulty in initiating sleep, either getting to sleep or staying asleep. So that is, not waking up during the night time for at least 30 minutes. So it took at least 30 minutes to get to sleep, or they were awake for more than 30 minutes over the night. And they had this issue for at least 3 nights of the week for at least 3 months. And they also had to have an insomnia severity index greater than 10. We excluded anyone who was currently taking any psychotropic medication, so anything basically that influenced their wake or sleep. If they were taking cytochrome P450 inhibitor medications, if they had a known sensitivity to cannabinoids or had been using cannabis in the past couple of months, anyone with any significant psychopathologic conditions, any people with current suicidal ideation, if they had another sleep disorder such as obstructive sleep apnea, a movement disorder, anyone who was consuming more than 2 standard drinks on average per week, or per night per week, or across the week. Anyone who's consumed too much caffeine, particularly in the evenings. People who were shift workers or athletes, or who worked in jobs that required them to have cannabis drug screen. Or anyone who was required to drive within 10 hours of taking the medication, because we asked people not to drive during the day. However, throughout the trial, that actually changed, and we were required to ask people not to drive for 5 days after taking a single dose.
We're going to actually hear more about cannabis and driving in this webinar, which is great.
All right. So our next speaker for tonight, Dr. Janet Schloss, was the lead researcher looking at the effects of medicinal cannabis on glioblastoma multiforme, which is an aggressive type of brain cancer with a very low survival rate, as you can see listed on the slide. The research is important because of the potential for medicinal cannabis managing either quality of life for patients or the progression of the disease. In this case, it was a randomized double-blind trial that ended up assessing 2 different ratios of medicinal cannabis, a 1:1 or a 1:4 ratio of THC to CBD, in addition to the patient's management plan. So let's hear some of the questions that we thought to ask her about this.
Hi everyone, here we have Janet Schloss as well. for the research on glioblastoma. And I'll start off with asking, before starting your research, what made you decide that you wanted to see the effect of medicinal cannabis on glioblastoma as a type of cancer in particular?
Yeah, it was really interesting. I was doing research on medicinal cannabis, and because I have a big interest in cancer, I was looking at what research had been conducted on different types of cancer, and really found that most of it had been done on glioblastoma, and that there had been, like, one of the first studies that they looked at for treatment of them, because it's got such a bad prognostic outcome. And then I was talking at an AIMA conference to the doctors, and Charlie Teo was actually there as well. He was presenting after me. So after that, I went up to him and had a chat. And I said, look, this is what I'm actually thinking of. I think that, you know, it would be worthwhile. And he said, yeah, let's do it.
That's awesome. And that would have been a big name to walk up to as well at the time, I'm sure.
It was. He's very lovely though.
Yeah, I know. I've only seen him on TV, I should say, at a particular show, but he seems so down to earth when you start to actually talk to him.
Yeah, absolutely.
So what was the sort of summary of results that you have observed so far for the trial?
Well, yeah, so we've now completed it, and we've got an ASCO paper, which was actually fantastic, and waiting for the manuscript to come out.
Oh, awesome.
Which is good. But we ended up with 83 people who completed, well, not all of them completed the trial, but were actually enrolled. And because this was a tolerability study, what we found is that everybody tolerated really well the two different ratios of THC to CBD. And we did a 1:1 and a 4:1, which was high THC.
Oops.
We had no major issues at all. We had no serious adverse events. The main side effects that we actually got were like dizziness, drowsiness, tiredness, dry skin, dry mouth. That was probably the main ones. We did have a slow titration up for it. And then when we actually look at the functionality of it, we found that it was actually statistically significant for both physical and functional for the patients themselves, but highly statistically significant for sleep, which was fantastic.
Yeah,
and I know everybody's also interested in the outcome of the tumours. We did actually do MRIs at the beginning, the baseline, and at 12 weeks. And remember, this is only like a snapshot. So we're still gathering survival data and all that type of thing. But what we found over that 3 months, remember these are people who've had a recurrence of glioma, is that 45% actually had stable disease or reduced growth. We had 16% that just had slight enhancements, and then 10% that had some progressive growth over that 3 months.
Wow.
So reality, that's actually a really good outcome.
Yeah, that's awesome particularly to be able to compare as well the scans and to see the percentages. That's really good results from my perspective. What would be something that you would have liked to have changed in the research that was conducted?
You know what? I actually wouldn't want to change really anything.
Yeah.
I like the fact that we did a pragmatic trial that was across the board. And yes, it wasn't a very, like, if you look at the medical fraternity, they very much like, you know, it can be this, it has to be this particular mutation segregated to these type of patients. Well, the hard part is with medicinal cannabis is we don't know who is actually going to work on the best. So it's one of the reasons why we chose a pragmatic study, so that when we can go on to do the next phase of the study, we can be a lot more specific. So probably the only thing I would have changed is that I didn't realise it was going to be so sought after, and that I would have put more people on the phones and getting things sorted when we had an influx of interest at the beginning of the trial.
Yeah, makes sense. And then now that you've finished up with this, what's sort of next in line for your research?
Yeah, so we're already in discussions with our next phase for it. And like we said, we're still waiting on the analysis of the cannabinoids and the endocannabinoids from the blood samples. And that I'm hoping that will actually help us to guide us to, which is going to be a much more specific inclusion criteria for it. But yeah, so we're looking at the moment at doing Australia-wide study, because one of the biggest problems, because it was only a single site study, was that people had to fly in, and that obviously caused problems with them because they have brain tumours. They have to increase dex, methazone consumption over that particular time while they're flying and afterwards. So to be able to reduce that and have like a trial centre in each of the main capital cities and maybe some regional ones as well, will be a huge benefit to the actual patients. And then we're going to be a lot more specific as to when we actually start and then what treatment it's actually used. Because we had a whole range of people who were actually on temozolomide. We had 30 people on temozolomide. We had 6 people who were on lomustine and avastin at the same time, and then 2 on avastin themselves. So we found that there was no interaction with any of them, which was fantastic. But at this stage, you know, it would be really good if we can actually target one particular type of drug in that people who are actually in that cohort. And that's what we're working on at the moment.
Yeah, that's really interesting as well, particularly for pharmacists out there with clinical drug interactions, and knowing that would be really good for, I'm sure, that patient clientele, but many others. So thank you so much for today.
No worries, it's been an absolute pleasure.
Thank you.
Alrighty. Sorry, Janet, for saying the wrong ratio. I realised it was 1:1 and then a 4:1, so apologies for that. All right, so our next speaker for tonight is Thomas Arkell, and he is from the Lambert Initiative at the University of Sydney. And he was the lead PhD candidate that conducted a randomised double-blind crossover design with 14 volunteers. And this trial looked at comparing the effects of THC-dominant cannabis that was vaporized, that compared to cannabis containing an equivalent amount, so a balanced formula of THC and CBD, and a third group of placebo on simulated driving and cognitive performance. The results are now actually published in the journal Psychopharmacology. So let's hear some of the things that Thomas had to say as to what they discovered.
Hi, Tom. Thank you for joining us today. Could you tell us what equipment you used in your study to assess driving skills?
Yeah, so when we're looking at the effects of cannabis on simulated driving, we have a simulator that we had custom built for us by a company down in Adelaide. So this was, it basically meant getting a whole lot of components from real cars, so things like the seat, the steering wheel, and stripping them out and then setting them into this metal frame. And then we mount these 3 big TVs on it around the dashboard. And so you get this sort of quite visually immersive 180-degree field of view visual scene. And then projected onto that, we have this software program that's running the simulation tasks. And so participants completed a mixture of highway and rural driving. There were sections that were designed to be demanding, difficult, other more monotonous, straightforward driving to see how people went when they were stunned under those different kinds of conditions. But really, yeah, the simulator was, I suppose it was designed to mimic real-world driving as much as possible and make participants feel like they were actually in a car, and with the haptic feedback and the tension you would normally get on the wheel as you were driving. And we also had a number of cognitive tasks as well. So these were computerized tasks, measuring things like divided attention, reaction time, processing speed, so things that are quite important and essential skills in day-to-day driving behavior.
So your study looked at vaporized cannabis, that is balanced THC CBD concentration versus the THC dominant. Could you tell us how oral formulations such as oils would compare?
So the main difference here really just comes down to the duration of effects. So with an equivalent dose of THC, you would expect probably a similar level of impairment with oral products. But the main difference is how long they last. So these products can often take an hour or 2 to kick in, and they may last for up to 6 to 8 hours, or in some cases even longer. So it's possible that that driving impairment we see with vaporized cannabis might just be extended. It might last for quite a bit longer with oral products.
Okay, that makes sense. And how would the impairment to driving with cannabis be different to driving with other CNS depressants such as opioids or benzos?
So look, in a way, it's a bit difficult to make this comparison because it really all comes down to dose and tolerance. But I suppose what's quite telling is there was a really big European study looking at the effects of alcohol and other drugs on driving that was published a little while ago now. And that concluded that the effects of cannabis were really fairly nominal. So that was sort of in the low risk category, similar to what you might expect to see in drivers, with a blood alcohol concentration of up to about 0.05, the legal limit. Whereas with benzos and opioids, those effects were a bit stronger. So they were more similar to what you might expect to see in a driver, with a PAC of 0.05 to 0.08. So it's a bit hard to say, that one's better or worse than the other, but I think the real difference is the sedation and the drowsiness that you can get with benzos and opioids you don't really have with cannabis, but they may present a protracted risk that you might not see with cannabis.
Right. All right. Thank you very much.
All right, and finally, I want to introduce you to our last speaker of tonight, Charlene Mavor, a medical dentist, and she's the Managing Director of Medicinal Cannabis Research Australia that was established in 2016. She is ideal to summarise what's happening right here, right now in our country. So let's hear what she had to say. And if you're still here and listening with us, after this will be probably the most interesting part of tonight's session, where we'll do question and answer time.
Hi everybody, my name's Charlene Mavor. I'm a medical scientist and also Managing Director of Medical Cannabis Research Australia. I've been asked today to give you a brief overview of clinical trials and research in Australia using medicinal cannabis. Medical Cannabis Research Australia is a charity that we established back in 2016 when we realised we needed to educate medical professionals and patients about the use of medical cannabis and also the endocannabinoid system. We also have a strong advocacy mission to try and help patients with access to legal medicinal cannabis products. And we also are very interested in advocating for research, promoting research and facilitating increased research into more conditions where medical cannabis may be used. So looking at the Australian research platform, we've got 69 patient studies which have been either announced in the media or are in progress. These also include patient enrolment in global studies. 11 of these have no trial listing as yet on any global trial register, including that of Australia. Of 58 listed trials, 7 have no ethics approval to date. 10 are using synthetic cannabinoids, but I want to talk about the other trials which are using whole plant extracted cannabis products. There are 20 non-commercial research groups conducting these and 29 commercial research groups. So with these trials underway using plant extract products, we see 9 safety and tolerability trials, 8 cancer and cancer symptom trials. One of these is you have already heard about, which is the glioblastoma trial run by Janet Schloss. There's also 4 epilepsy and seizure syndrome trials. Looking at chronic pain, we have 5 trials. Sleep disorders and insomnia, 5 trials. Looking at medicinal cannabis and its effects on driving, we have 4 trials. Of course, this is a very important research topic and we need this information out as soon as we possibly can. There is also 1 behavioural disorder trial. And further to that, there's 11 other indications trials underway. And I'll quickly run through those. Tourette's syndrome, prevention of acute graft versus host disease, amyotrophic lateral sclerosis or motor neuron disease, gingivitis, inflammation, muscle damage and functional recovery after exercise, anxiety disorder trial, a dementia trial, an alcohol withdrawal trial using cannabis, Brett's syndrome, and also there are a few observational studies underway or about to be underway looking at a multitude of conditions and products. Looking at international trial involvement with Australians, GW Pharma has spearheaded most of this, and this is through their research arm, GW Research Limited. So we have a study to investigate the efficacy and safety of cannabidiol with Dravet patients, and we have 2 clinical trial sites in Australia, in Victoria and New South Wales. This trial was actually just completed, so hopefully we'll be seeing some results and conclusions written up in the literature very soon. There's also 2 open label extension trials using cannabidiol, one for seizures in tuberous sclerosis complex. 12 children in New South Wales are participating in this one, and this is ongoing, so results to follow. There's also a trial for Rett syndrome, and we have patients in Perth and South Brisbane, Queensland participating, and that is due for completion September next year. Australian trials and studies. We have already heard from the Lambert Initiative and Ian McGregor regarding their Tourette's syndrome RCT. There's also 2 trials which will be observational and also looking at multiple conditions and multiple products. The first one is being run by Camberlake Clinics and Swinburne University of Technology, enlisting 2,000 patients, looking at dosage patterns and efficacy, and also an upcoming trial called the Cannabinoid Medicine Observational Study, and that will be looking at enlisting 20,000 patients, so over a number of years, and that is awaiting ethics approval. So that's been run by Applied Cannabis Research and Cannabis Access Clinics, and I know they are using Little Green Thumb products, but possibly other products as well. Now, I find this a bit of an exciting Australia-wide trial, which has been proposed. Cannabidiol as a treatment for young people with ultra-high risk for psychosis, proposed as an RCT, and this will involve 405 patients, and that is being run by Origin with the Wellcome Trust UK grant. That's enough from me for today. Thank you very much for your participation. And please do follow us on the socials, where we do continue to share Australian news about patient access, as well as concentrate on sharing research and upcoming research in Australia and from around the world. Thank you very much.
All right. So I'll now invite all of the panelists from tonight. Thomas won't be joining us, but Ian McGregor is from the Lambert Initiative, and you can light up your video cameras and microphones, and we'll try and go through some of the questions. So let me just stop sharing my screen. Everyone's here. All right, I'll start going through some of the questions. If the viewer, still has one that you would like to pop down, just use that question and answer button rather than the chat button. So for the dementia trial, Amanda, I might quickly check with you what strength of the product we're using, or what ratio of the THC to CBD.
We're using a 3:2 ratio, so a higher amount of THC compared to CBD. So it's a 3:2.
Awesome. Okay. And someone else has asked as well, what regimen did you use for that trial for dementia? So I think maybe they mean how often per day or night.
So for the first 2 days, they get it once a day in the afternoon. So about 2 p.m. is the first dose for the first 2 days. And then they get it twice a day from 8 a.m. And then also again at 2 p.m. with the dose increasing on the 2 p.m. dose on days where it is increasing.
Okay, cool. All right. I'll go to the insomnia trial. I don't know if Jen is around. Hey, Jen.
Hi.
It's a similar question in terms of strength. I guess if you could cover what it was for those patients and at night, I'm guessing.
Yeah, sure. There's a couple of questions that I've already answered through the Q&A about this.
Okay.
The formulation is actually proprietary, but I can tell you that it's a combination of THC, CBD, and CBN. And yeah, as you've already said, it was taken at night time, about an hour before they were expected to, or they were planning to go to sleep.
Okay, cool. And we can certainly look at sending out a link as well with some of the research that has already been published. But if you're savvy enough, you can do it yourself as well, like I did before this. Let's see what else we have. Dr. Igor Yakubovich has asked the combination of THC and CBD for glioblastoma. What was that again, Janet? Sorry, I've confused everyone.
Oh, no, that's okay. We compared a 1-to-1 of CBD to THC to a 4-to-1 of high THC to CBD. And our 1-to-1 was basically just around 5 milligrams per milliliter for both, just slightly under. The 4-to-1 was basically 15 milligrams per milliliter to 3.8 milligrams per milliliter.
And then the results from one or the other, what did you find with tumour reduction?
With the tumour reduction, what we actually found was we didn't actually compare that because, like I said, it was only a snapshot. So we didn't have efficacy as our primary outcome. It was actually tolerability. But we did actually find with their quality of life information that the 1-to-1 was actually better than the 4-to-1. The 4-to-1 was actually better, was actually found better for pain in a range of different areas, but the 1-to-1 was the majority.
Okay, cool. I might ask this next one for Charlene actually, because I think you would look a lot into this, whether studies are using isolates, so CBD or THC, or a combination of the two molecules as full plant extracts. I guess what do you tend to see with research at the moment, and why do they choose one or the other?
So your question was, are there studies using isolates? Was that right?
Yeah.
There's not very many that I've seen in Australia. There's a few of the commercial entities are doing some synthetic cannabidiol studies, but mostly I'm seeing their whole plant extracted product studies at the moment with what's happening in Australia.
Yeah, I agree, commercially as well. Does anyone else have anything to add, I guess, with the trials that you decided to start and whether you chose an isolate or not, and why? Not really?
No, I'm happy to talk.
Yeah, sure.
I think that we did a review leading up to it, and the isolates were not as effective as the whole plant extract. And what we did is we had Justin Sinclair on board as an investigator, and he looked into it very closely. And that the most of benefits is that we look at CBD and THC, but reality is there's like 140 different cannabinoids and other components, over 300 different components of medicinal cannabis that we still don't know the benefits of. And in an isolate, you don't actually get that whole component. And I think that when we're looking at what is of benefit, we need to actually consider it as a herb and a plant that has a whole synergy and that entourage type of effect. And we still have so much more to know.
Totally agree.
Yeah, that's very true. Particularly with the literature out there with that synergy behind it. There was another one for yourself, Janet, in terms of, can you comment a little bit more about the importance and the benefits of multidisciplinary cannabis research in cancer, and Do you think that the quality of life and the sleep benefit is product and or cannabinoid or terpene specific, or if it's more general cannabis specific?
This is a really interesting cohort that we're actually looking at, and we have a high majority who are actually on dexamethasone, which obviously affects their sleep in general. And I think number one, the first question that was answered was, is it important for multidisciplinary? Absolutely. I actually think that it's imperative that we have a whole different range of teams with different specialities and professions to be able to help different cohorts. And that does not just for GBM and cancer, it goes across like all different chronic, particularly chronic health conditions. And I think that the more that we can work as multidisciplinary, the better it will actually be for the patients. And for me, it's always patient-centered. It should always be patient-centered, moving forward. So if we look at what is most of benefit, do I think that it's the cannabinoids? Do I think it's actually cannabis that's actually helped with the sleep? It's really hard to sleep. Jennifer's better to answer the sleep question than I am. But we had it was highly statistically significant with 0.001 for sleep that they had the benefit of. But we did a single dose, basically an hour before bed. And it could be that the THC had a component. We found the one-to-one just worked just as well. And it worked for both getting to sleep and staying asleep for them. So at this stage, we don't know. And like I said, Jennifer's probably better to answer the sleep question than I am.
All right. If you want to add on, Jennifer, that's fine. Go for it.
No, I think that the evidence so far, there's not been enough trials really to say definitively that yes, it does assist with sleep. However, there is a lot of uncontrolled studies and a lot of anecdotal data which indicates that it does benefit sleep. In terms of the ratio and which cannabinoids, as you say, there's so much work to be done. I'm curious, Janet, how were you quantifying sleep? What was your metric? Was it self-reported?
It was self-reported. So we had a number of different questionnaires that we were actually using, and we had a daily diary that we did, but we didn't have a band or anything that was actually recording it like yours was, because it was just a secondary outcome versus a primary outcome.
Yeah, and so a lot of the understanding of the benefits on sleep, or poor sleep insomnia, is from trials exactly like yours, where it's a secondary outcome measure. Yeah.
All right, let's hit the topic of driving now. So Ian, thank you for joining us for tonight. Have you had results from the driving study influence some of the Australian driving regulations, particularly in relation to THC levels?
Not yet, and forgive me, I don't have video tonight, so you've got you've got a picture of a qual and sticker. I hope you like quals. Yeah, I was surprised actually you didn't ask Tom about his most recent study. He's been over in the Netherlands for the past year.
He commented on that at the end, sorry. But we had
but simulator studies are great, but where the rubber really hits the road is when you've got people in real cars on real roads, and that's just exactly what he did in the Netherlands. It was a very adventurous study, and so one of the major issues there was whether CBD, vaporized CBD, actually affected driving in the same way as THC, and it'd be no surprise, I think, to this audience to find out that CBD didn't impair on-road driving at all. With THC, we did get some impairment, and a major issue we were trying to resolve with that study was duration of impairment. When you talk to patients, they really want to know, how long should I wait after taking my medicinal cannabis before I drive? And so we went after that quite deliberately in the on-road study, and we found that at 4 hours, basically the impairment we saw at 1 hour had dissipated. And that was with THC only cannabis, THC CBD 1-to-1, and then with CBD dominant cannabis, we saw no impairment whatsoever. So we hope these results will provide some clarity to patients, but also perhaps impinge on I guess, the regulatory space, the legal space around cannabis and driving. I think Tom made a very important point as well, that if you take oral products, there's also the possibility that there'll be an extended intoxication with THC beyond 4 hours that we see with vaporized. The other issue that we're going to go after in our next study is whether you get tolerance to driving impairment. And there's already a little bit of a literature suggesting that if you're a habitual user, then you're a lot less impaired than an occasional user. And all the work that Tom has done to date has been with occasional users, where we see quite clear impairment in terms of their driving capability for up to 3 to 4 hours. So these are important things that will feed back into, hopefully, the regulatory space. But at this point in time, I would recommend to patients, and we get asked this all the time, how long should I wait? I'd give it at least 4 hours. If you're using oral products, maybe 6.
Okay. And I might wake up Professor Marco Falasca and ask you, I guess, what the next plan is for you with the human trials, if that's going to be happening here in WA, in terms of what you've discovered from doing it in vitro.
Yeah, we hope so. I think we are, for the data we had so far, we are ready to start. It's, of course, there are many things we are now, there are many complexities, starting from the dosage, the combination, try to find the best combination. So, of course, this takes time because you need to taking the long way because we need to do a proper study. And but I think we can start. I hope we can start very soon.
Awesome. Yeah, I hope so too, because it will be really beneficial.
Yeah, of course, this is, we all hope. But I think with we have some clear response by now, and with this clear response, I think we can move forward. Okay. Obviously, COVID is not helping us, but but yeah.
Are you looking at it as well in terms of the aspect of cell endocrine system and cell metabolism with diabetes? Because I've just finished the diabetes educator course, so we didn't actually touch medicinal cannabis at all as a topic in our course. So
yeah, we are investigating on that aspect, and of course, compared to the cancer field, we are a bit behind, but it's also very interesting in that.
Yeah, definitely. Yeah. Okay. I might go through another question from the question and answer button. So someone's asked if an indica strain of legal cannabis oil is recommended for best sleep and helping with insomnia rather than sativa dominant type of cannabis extract from the oils and products that are out there on the market. Sorry, that question kind of confused me, sorry. I think what they're trying to say is whether one strain is more beneficial than another. Jen, sorry.
I think that I've got a lifetime of research to do to answer all these questions about strains and formulations. I think there's so much we don't know. So the answer, unfortunately, is it's not been done. So
yeah. No, that's okay.
Interesting, because what the question was is that was indica in any of the formulations. That none of the ones that I know of actually have indica. It's all based on sativa.
Yeah, that's right.
The black market uses indica.
Yeah,
and that's where we also see issues. So there's still a lot of research to be done.
Yeah,
and it's kind of interesting as well that we don't tend to find out terpene levels, and it's not something that's reported. But obviously, we're going to have different terpenes in indicas and sativas, and that's where we're finding the big differences. So, yeah, maybe in future years we'll see this kind of come to the forefront a bit more, and I would love to see more research or knowing what terpene levels are in research projects going forward.
Yes, the terpenes were measured in our trial. I actually can't tell you what they are. Off the top of my head, I can't remember. But yeah, again, that's proprietary, unfortunately.
Yeah.
We're measured actually in ours as well, so we actually have them.
Great. Well, that's really
We
great.
have the different components.
Because I'm sure that would, you know, help base
what conditions you'll be looking at, knowing that information.
Marco, I had a question come through my email, and it was asking as well whether you've been approached by patients using cannabis already and on chemotherapy, or whether they're considering it, or
Yes, many. I received many inquiries from patients, and I know there is using. I always suggest them to do it with approval of the clinician, the oncologist. This is obviously the best way forward. Not always the oncologist supported, but I know there are good response. Of course, these are negligible. These are not proper trials, but there are very, in pancreatic cancer, if you have a positive outcome, it's unfortunately not so common. So therefore, it's meaningful. But of course, we need formal scientific proof with the trials. But yes, there are many patients that are, especially for pancreatic cancer, that are interested in using cannabinoids or cannabis.
Yeah, I agree. We get a lot of phone calls as well from patients here at Health House, where sometimes it's even their family members calling up and saying, look, I'm too scared to tell the doctor, and should I, shouldn't I, should I stop, should I start on a higher dose? So yeah, it's very common. Amanda, someone did ask as well whilst we were chatting if you decided on using the 3:2 ratio of the oral spray for your dementia trial, or did you get to choose that because of research that you thought was appropriate?
No, that was decided by our industry partners. So the more from the lab where the production stuff is grown and manufactured. So it was a product that was given to us to then test. So we had no say from a research perspective what that ratio was going to be at this stage. Yeah.
Okay, cool. And that was only conducted in WA, is that right? Are you thinking to spread it to other states potentially later down the track?
Yeah, currently it's only in WA. We do have contacts over in Melbourne and stuff that we're hoping to expand to at some stage. Yeah, we'd love to go national if we have that opportunity.
Okay, awesome. I might go through one more with Ian. I'll bring you back with a question on impairment. Someone has asked if, so they're asking with administration or the use of short release THC with methods such as vaping, if that was potentially less impairment using a slow release, if it would be a potentially less impaired patient if they were using an oil, so a slower release type of product. I think Tom mentioned in his video a little bit about this and the onset and duration.
Yes, that's right. I mean, basically, if you vaporize or smoke, you get a very sudden spike of THC in the bloodstream in the brain, which does give rise to impairment. If you use an oral product, you get a kind of ramping up of THC in the bloodstream, which peaks around about 2 hours, but lasts a bit longer. It's got a longer tail. So there's an interesting difference there with route of administration. Yeah, the other thing, though, I think to say about cannabis and driving is the subtlety of the impairment. So we see a moderate increase in what's called standard deviation of lateral position, which basically the amount that you weave on the road. So there's a little bit of an increase in weaving, which is equivalent to, say, about 0.05 of alcohol if you have a pretty big dose of vaporized cannabis. But at the same time, unlike alcohol, patients on cannabis and people on cannabis tend to leave a larger distance between themselves and the car in front, and they also tend to drive with less speed than people who are not on cannabis. So it's almost, I think what we know is that people on cannabis have insight into their impairment, and they tend to compensate for that. And this tends to flow through to epidemiological studies of crash risk, where compared to alcohol, the effect on cannabis on crash risk and crash culpability, the chances of being responsible for a crash, are actually very modest and sometimes statistically can't be discerned. So there's a lot more to learn. And just like Amanda, you know, with her sleep, I think we can do driving studies into the future. Sorry, Amanda, with her dementia, and also the studies with sleep. There's enough scope to just keep on going with all sorts of different routes of administration, different cannabinoid profiles. There's an awful lot to learn, and it's so important, this work.
Definitely. All right. Well, we've got another one that's come through. Let me just find what I thought was interesting. CBG, has there been any research on the benefits of this one, Charlene? I might start with you, but I think
I was actually going to reply to that. Please.
Oh, sorry.
But I'm sure, I know that there has, obviously preclinical, but I can't tell you any details. I just can't remember at the moment, but yes.
All right.
I'm sure Ian's got something to say though.
I could go on all night about CBG, but you know, there's this phrase, CBG is the new CBD, but it isn't. It's very different in a lot of ways to CBD, but it's strongly anti-inflammatory. There's potential effects on anxiety. In some of the animal models we're seeing, it actually does opposite things to CBD and has a very different neural receptor profile in terms of what it hits. There's probably a niche for CBG, but it certainly isn't the new CBD. It's something completely different. I'm aware of phase 1 trials with CBG that are going on in North America at the moment. So this is first in human trials of CBG isolate being conducted by a colleague of mine. So once they report, we'll know a lot more about CBG in humans.
All right. There's so many other molecules as well, so we could go all night, but I'll have to stop there. Thank you so much to everyone for joining us for tonight, and particularly the panelists. Thanks so much for taking your time to even record with us before all this, and you all look wonderful and the same as the original videos. So what I wanted to mention was we have recorded this and we'll put it up on YouTube as well, and we'll send you out all the link after this. And our next webinar is actually going to be on patient stories, same time on Wednesday night, and that's on the 12th of August. So keep your eye out for that in the emails. And I just want to say again, thanks for everyone for joining us, and have a good night.
Thanks, Donya.
Thank you.